Huh.
Okay, sorry guys, I guess I was on mute. Can everybody see my screen, even if I need to go back? Hello?
Yes.
Looks good here.
Okay, and you can hear me too, right? Okay, good, because otherwise you're just gonna see a guy that's gesticulating. Okay, so starting last year, so this is what's been happening, right? So we had this record-breaking rainy year in 2017. Apparently was the wettest year on record in the history of California. And we forget about it, right? Because our short term. And then we entered this new dry cycle. And now that we are potentially in this mega drought, we're seeing extensive tree mortality. And the tree mortality, I researched extensively, it's mostly in the area, and it involves exotic species. But I have to warn you that at the end of the presentation, I will present some data that shows that we have very similar pattern of mortality happening on native species. And I'll explain a little bit why we're seeing this mortality in these particular native species. And I think that the take-home message is that there is a common thread among what's happening to all these different tree species. And I wanted to acknowledge the SFPUC, which is your neighbor, who funded most of the research, the Forest Service that coordinated the research, and also the East Bay Regional Parks, which amazingly actually funded some of the research. And I'm very grateful to Pam Bites. She really was instrumental in getting us some funds. And also wanted to warn you that this research was done in a very short time because the agencies wanted to have they wanted to know really fast what was going on, and which is not unusual, but it's unusual for a UC lab to actually agree to do that. So we stopped all research, we stopped all other projects to work on these projects. And so as a result of that, we actually were able to get some interesting and fairly convincing results in a very short period of time. But remember that we should still consider these results as preliminary, because the sample size is good, is robust, but it's not particularly large. And I wanted to thank three people from my lab: Ines Marquez, Tina Pupinak, and Doug Schmidt, that really wore those Tyvek suits for weeks and weeks, doing all of the field work, and then continuing with the lab work to identify what was going on. And so the objectives of the study that we just completed about 2 weeks ago, and that we started in December of last year, was to understand what the drivers of large-scale dieback of both exotic and native species that has been reported in different locations in Northern California. And I will show you some maps, but mostly what I will be talking about is eucalyptus and acacia mortality. So immediately you realize that we're talking about mortality of trees that are exotic. And I think we understand now that these trees were not planted necessarily in areas that are ideal for them. They were planted with the false pretense that they're drought tolerant. It turns out they really are not. I mean, at least acacias are not particularly drought tolerant in their native area, in the region in Australia. And eucalyptus have an appearance to be drought tolerant, but in reality, the reason why they can do okay with little water is because they can suck up the water much better than other plant species. So there's been issues with these trees really depleting the water resources where they're planted, even if they, until now, they seem to be doing okay. And then the other tree species that I'll mention at the end of the talk are tree species that are either particularly mesic, so they're tree species that originally should be riparian near rivers, and because of fire exclusion, they have now spread throughout woodlands on the coast of California. And I will also talk about a tree species, and I should really talk about two tree species that grow in a very harsh environment on the Sierra foothills, where the temperatures are incredibly hot at the low elevation. So these are all tree species, all of these, including the exotics, that probably are experiencing some issue with the climate and the ecology of where they're growing. And so we were asked to find out if there were any general symptoms So was there something, you know, in the appearance of these trees that was common among different locations? Were there signs that were common? So when I say sign, I refer to structures that are produced by pathogenic fungi. And then did we find fungi that were consistently isolated from multiple or all study locations? So the idea behind the study was to look at a significant number of study sites with multiple trees per site, intensively sampling each tree. So each tree took, you know, hours to sample because we cut down the tree, we looked at the foliage, at the branches, at the stems, and so forth. And the big question was, okay, are these trees just dying or dying back because of climate issues? So is this drought? Or are there fungi that are involved? And in order for a fungus to be involved, we would expect to find this fungus in all different locations. Like, you know, now that we know about sudden of death, where we see large scale tanoc mortality, we go and look for the sudden of death pathogen and we find it. So basically, a consistency of presence would be a very good indicator. And then we would confirm the role by doing, you know, inoculations where we take healthy plants and we see whether we can recreate the symptoms. Are there secondary fungi? So these are what we call opportunistic fungi, and these normally come on to hosts when the host is particularly compromised. So when the health is compromised, then these fungi have a chance to attack these hosts that otherwise are unavailable to them. And some of these secondary fungi may be rare, or, and some of them may be site-specific, but significant in a site. They may play a role. So some of these secondary fungi may actually be more aggressive than we thought. And so they may hasten tree mortality. So we wanted to test that as well. And then the big question was, of course, there was some, you know, chatter about maybe a new exotic pathogen that was killing all of the acacias and all the eucalyptus. And so the big question that the agencies wanted answered, is it so? So is it a fungus, and is this fungus native or exotic that is causing this mortality? So this gives you an idea of the scale of the mortality. On the left-hand side, you know, I don't want to spend too much time. I don't have the same map for the peninsula, but basically wherever you see colors overlaid on top of the map, there is some level of mortality, just different species. So as you can see in the East Bay, it's really large. You can see color all the way on the Carquinez Strait and then all the way down to the southern part of the bay. And if you went and looked at these trees, I think these are actually bay trees that we're looking at here, you would see these trees dying back, so browning and desiccating. I should also say that both with the acacias and the eucalyptus, and maybe even with the bay laurels, it's not really clear whether every tree really is dying or whether it's just the canopy is just browning and it's going to resprout. At least in some cases, the roots are not compromised, especially for eucalyptus. So there is a chance that these trees are not really dead, but they're just dying back, and they're going to have a chance to grow back. But we'll talk about that more in general. And so here, I wanted to give you just a quick overlook of the study sites. So we had 6 sites for eucalyptus, and we had 4 sites for acacia. And I don't know if this is wrong, this is wrong or not, but we actually had, let me close this because, so actually it's the opposite. So we had 4 trees per site in eucalyptus and 8 trees per site for, 8 trees per site for acacia. So this is inverted. So we compensate a little bit, but as you can see, the site is fairly well distributed across the Bay Area. So they cover a fairly large area. And each one of these sites, each one of these dots represents multiple trees. Again, 4 for eucalyptus and 8 for acacia. Sorry about that mistake. This is what the mortality looks like for some of you that may not have seen it. This is probably the worst, visually the worst site. So this is Blackwood acacia. As you can see, a very large patch of acacias that are dying back. So this is what we were working with. And once you get closer, remember, we were actually trying to find symptoms that were common. And in the acacia was immediately clear that there were these fissures on the trunks of the acacia. And we suspected immediately that there was a canker fungus. So a canker fungus is a fungus that kills the tree. It's a little bit like the sudden of death fungus. But the sudden of death fungus is so aggressive that the plant doesn't even have the time to respond. Sorry about that. So we don't really get these open lesions that are visible in this case. In this case, you can see the open lesion, especially in the second one from the left. The tree is actually trying to respond to the cankering. You can see the smooth margins of the lesion. But in any case, we saw something that was fairly common across all sites. Now, of course, when trees are dying, there are literally hundreds of fungi that are contaminants. And we have the secondary fungi that I was mentioning. So the big issue here was to identify the perfect type of tree to sample. And the perfect type of tree for us is a tree that shows symptoms, but it's still green, and it's not just a little bit green. We want the tree to be a little more than just a little bit green. So we want the tree to still be alive, but we want to have clear symptoms. So those were the trees that we were focusing in. We were not doing a random sampling. So we're not just sampling random trees. We were specifically targeting trees that had these symptoms. In the case of the eucalyptus, so we'll talk about these first two species together because it's a similar and different story at the same time. This was the way that these eucalyptus stands were looking. A little bit similar to the acacia, although eucalyptus are kind of brownish sometimes on their own. So it's a little bit more, it's not as stunning visually speaking. But basically what you're supposed to see here are the crowns of these trees are drying out. They're thinning on top. You can see that, especially on the right, you can see the tops are thinning. So there is premature abscission, a drop of the leaves, and there is dieback in these branches. But in eucalyptus, we didn't really have those open fissures on the trunks. So we really didn't have any obvious major canker on the stems, on the boughs of the tree that were reoccurring, suggesting that maybe the true trees were experiencing something slightly different. And indeed, when we started cutting into the acacia, there were multiple symptoms. Okay, so the symptoms were not all identical, suggesting that there were several fungi involved in causing disease on these acacias, but there were some, like the ones that are here, that were fairly recurrent, and especially the ones on top. You see this pie-shaped dark lesion. This is not chainsaw, burn. This actually is. You can see the bark is cracked on top, and this is a lesion. And I can tell you that it's a similar lesion is to the right here on a smaller branch. And then we would have these lesions at the root collar as well. And this probably that you see here are actually caused by two different fungi. But these were very strong symptoms that we were seeing inside the acacias consistently, even if, as I said, it was a multitude of symptoms, but we were consistently seeing them. And the pie-shaped lesions were very, very consistent. And these lesions on the root collar were actually fairly consistent across sites. When we look at eucalyptus, the consistency was not there. The only consistent symptom in eucalyptus was leaf blight. And so I think I'll have a couple other pictures, but here you see this. Oh my God, sorry about that. The only consistent symptom was the browning of the foliage. The browning was sometimes it was heavy spotting and the spots coalesced in larger lesions. But that was fairly consistent. And then, and that's why I put this arrow here. And then we had other symptoms that were site-specific, including these lesions in the branches and sometimes in the stem. And there was some bleeding associated with these lesions. And the bleeding made us think immediately that maybe the specific fungus was involved. And I'll tell you about this fungus very soon. But these symptoms that you see here on the right-hand side of the foliage were definitely site-specific. They were not ubiquitous in our study. And so we actually went, we were asked to, the funding engine said, you got to give us a report like now. It takes weeks to actually do all of the study. And so we had a major, major effort here was actually done to isolate. So actually, what does it mean? We actually wanted to grow these fungi in culture, and we ended up with about overall 1,000 different cultures here. And what you're seeing here is our morphological grouping of these plates. So to analyze 1,000 plates would take us a year or two, and that was not acceptable. So what we did, we generate groups that to us looked identical. So each pile has identical morphology, and here is one pile that's shown to you. So you can see on the right-hand side, you see that those are fungal colonies growing on a growth medium, and you can see they're very similar, right? So they are whitish, and then they get yellow color. So our assumption was the one group would be one fungus, okay? And then we did.
He breaks it, he pays for it.
Yeah, if you can mute yourself, I can hear you. So the cultures here on the right-hand side were expected to be the same. And so what we did, we took 2 or 3 of these, and we actually confirmed they were the same by DNA analysis, okay? And to be, actually, I complimented my folks because in the lab, they were 99% right. So normally when they put things in a pile, they actually, based on DNA sequencing, they were the same organism. And so what did we find out? So this was a result that combined, culturing, observation of symptoms, culturing, and then DNA analysis. And so we collected 81 wood samples or leaf samples from acacias from 30 trees, and 124 samples from eucalyptus from 24 trees. And you've seen the different sites. For acacias, we had the stem and branch cankers that were visible at each site. In eucalyptus, it was only foliage browning and twig cankers. In acacia, we also had other types of wood discoloration, and they were, they were not necessarily site specific. Sometimes they're present in 2 sites. While in the case of eucalyptus, we had some very specific signs and symptoms of advanced, advanced disease or incipient advanced disease, like heart rot. We had branch cankers, we had sapwood staining, we had mycelial mats, which is we could actually see the fungus growing under the bark. These were mostly site-specific, and they're usually indicative of a tree that's really on its way out, but they were not broadly present. And so we knew that these were not really major drivers of what we were seeing. They were more secondary drivers. So what were the things that we saw everywhere? So remember, our hypothesis was, okay, if something is really driving this mortality, we should find it everywhere. And then we should have a hunch that this is actually a pathogen, because there could also be a contaminant that's everywhere. It's not really significant, so you got to have that knowledge. So when we excluded all the contaminants, like things that we know are in the air, in the soil, and so forth, and there were hundreds of those. So it was meticulous work. We actually found 2 fungi in acacia that were present on almost every site and on every tree that gave us isolations. So some trees were just too old, the disease was too advanced. We didn't really get anything but contaminants. But on every tree that gave us some good fungal isolates, we either got Diaporthy funiculina or we got the Diphthorella viticola. And often, most often, only one of the 2 was found. So either we found Diaporthy funiculina or we found the Diphthorella viticola. So now these 2 are, they are already known to be in California. They're already known to cause disease. They're both relatively broad host, so they can affect a range of different plants. So the assumption was that these 2 fungi, jumped from other hosts to acacias, and then started causing disease on acacia. So basically, if there was an inoculum source at some point, and then acacias tend to grow in very dense stands in general, And these dense stands have no social distancing. And because of the lack of social distancing, infection between trees becomes very easy. So there's a lot of contagion. So this would explain the ubiquitous presence of these two, knowing that they are reported to be fairly aggressive pathogens. And the origin potentially is California. So California provided this pathogen. So remember that acacias are not native. So this is a combination that is potentially very deadly because we have a fungus and a host that have not co-evolved. On the eucalyptus, we didn't find anything that could really cause tree mortality, like Diaporthe funiculina, Dophyra viticola. They can cause lethal cankers. The only thing that was ubiquitous was foliar pathogens. And both of these are foliar blights. They can go into the twigs, and in fact, we did find them into the twigs. And the most common one was Pseudosidova eucalypti. And then we also found Claudosporium in almost every site. So we have two foliar pathogens that would really explain why the browning of the canopy was so widespread in eucalyptus. But again, we didn't have this canker fungi. Ooh. There were some interesting things. In Acacia, we found one zygomycete that was present in trees that were obviously dying, and we didn't find Diaporthe funiculina or Dophyra viticola. And these zygomycetes are very common. Normally, they're considered beneficial, so they actually consider part of that mycoflora that makes the soil rich. But this particular one, Umbellopsis ramaniana, we actually found it causing symptoms. So very similar symptoms to those caused by Diaporthe funiculina inside the wood, only one site in the peninsula. And so we thought, well, is it possible that maybe this fungus too may be causing mortality? So maybe this may be a primary player, but only in the site where it's present. It wasn't, as I said, it wasn't ubiquitous. In eucalyptus, that was not present. But what's common about all of these pathogens that are listed here, except for the last one, for the zygomycete, is that they are, but also the zygomycete actually, they're all known to be endophytic. So they're all known to be fungi that can live inside a tree without causing any disease or any symptoms for very long period of time. So from from years to decades. And normally, they actually coexist within the plant tissue, it seems, by losing their cell wall, for those of you who don't know. So they lose their cell wall, and their cytoplasm becomes part of the plant cytoplasm. And this discovery was made this year. So we didn't know until this year how it was possible for these fungi to actually coexist in a plant without causing symptoms for decades. And this is an hypothesis which was well proven by experimentation. And when the cytoplasm is included in the plant cytoplasm, actually the part of the gene expression of the fungus still works. So basically the plant and the fungus express their genes together, sometimes using the same machinery. And this is why normally these endophytes are actually considered as being potentially beneficial. However, if these fungi are, if there is a physiological trigger, so if the plant goes into some kind of stress that normally involves a reduction in water availability in the plant and an increase in gas, then these fungi switch from being beneficial or neutral to actually being pathogen. And in the case of the Diaporthe funiculina and Dophyra viticola, they can be deadly lethal pathogens. And in the case of Pseudosidovia and Claudosporium, they can be very aggressive defoliators. So the point here is that these fungi being inside the plant already, they're very fast. And this is why if we had a very wet year in 2017, how come in 2019 plants started to die? It didn't really make sense, even with a 2-year drought. We know that these trees have gone through longer droughts without displaying this large scale mortality. But if we put in the equation the activation of the pathogenic phase of these fungi. So these latent pathogens, once they're not latent anymore, they can be very, very aggressive, and they do their job. And in the case of Diaportha dothrella, they can kill the plant. And in the case of Pseudopseudophila, they can kill the foliage. So this was consistent, and this was a common thread. But obviously, it was a different common thread, because killing the foliage and killing the entire plant is different, at least the aerial part of the plant. And the other big difference is that Diaportha funiculina, and this is an important one, and Dothrella viticola are generalists. So there is a risk that the buildup that they're experiencing on acacias may result in infection of other plants that are in stressful, conditions, and potentially native plants. So there is no reason why native plants could not be affected by, sorry, I went backwards, by these pathogens. In the case of Pseudopseudophila eucalypti, this is very host specific. So we know that this doesn't come from California. Pseudopseudophila eucalypti came with eucalyptus probably from Australia, and because it's not officially present in California, but everybody that works with eucalyptus knows about this fungus, because everybody eucalyptus is grown, you always have these symptoms when trees become particularly stressed. So do we have any sign? The big question was, do we have alien pathogens that we really should worry about? Okay, in acacia, we had only one such alien pathogens, and it was a relative of Dothrella viticola. So Dothrella viticola was one of the big players. But in one site in Oakland, there was also a relative of that Dothrella, which is called Dothrella monetae. And Dothrella monetae is unknown to California, and it's very, very specific to acacia. And it's also one of those latent pathogens, right? Because it's the same genus, so they all function more or less the same way. And so we think, yes, this was brought into California. Is it a real risk? Well, it's specific to acacias, and it's only present in one site. Well, I guess probably it's compounding the mortality in that one site. So it is making a difference locally, but in my opinion, doesn't represent a threat to the California flora being so specific and limited to a one site. We did greenhouse inoculations, and yes, in small potted plants, it was slightly more aggressive than Dothrella viticola, and it seemed to be more aggressive without the physiological stress. But honestly, on a small plant, the amount of disease that it caused was relatively small, and so it was difficult to gauge whether it really, it can really be more aggressive than Dothrella viticola, which is the California native fungus that jumped on acacias, and it's known to cause. So Pseudopseudophila eucalypti is, as I say, it's endophytic. It's present everywhere eucalyptus grows, so I'm not particularly worried about it. Neophysicarpum eucalyptorum is reported in California, and we found it in one site. So again, we don't think it's a major player. It was causing cankers on the branches of eucalyptus in one side, but again, because it was so limited in the distribution, we don't think it was a major player. So basically, my conclusion is that we don't really have alien introduced fungi that are major players in what we've been observing. And some more pictures of what these fungi look like. This is the analysis that shows you the different. So this is for acacia. You can see the two big ones, the two biggies that were present everywhere are shown there, Dothrella viticola funiculina, and then you have the relative that's only been reported from Australia, which obviously we now have in California as well. And we already talked about these. And so I wanted to show you a little bit without going through this. This is the hosts. So the only one that's exotic, it's there, but Dothrella viticola and funiculina are present in multiple countries, including California. And you can see the host range is pretty large. And so we're a little bit worried that the acacia outbreak may result in further outbreaks on other species that are not necessarily acacias. There are other fungi that we thought may play a role in hastening mortality. these were only found in trees that were already infected by Diaporthy or Dothrella. So this is, in Acacia, was very interesting. Our hypothesis really worked. And so these, we feel comfortable that are secondary fungi, and I won't talk about it, except for these Umbellopsis, that it was actually found in trees that were not infected by Diaporthy or Dothrella, and these trees were actually dying. So we did have the question of Umbellopsis. And so we tried to answer the question of Umbellopsis, and we tried to prove that we were right. And here on the left, well, I'll just start showing the one on the right. This shows you these the virulence of these different pathogens in natural conditions. So these are in trees that are standing in the SFPUC. We picked healthy trees and we inoculated them. And the higher the bar, the more aggressive the pathogen is. And so we were able to prove without any doubt that Diaporthy is definitely a primary pathogen, very aggressive, to be concerned about. And we were also able to prove that Dothrella and Umbellopsis were pathogen. And obviously Umbellopsis is a new finding because it's never been reported as a plant pathogen, but you see the amount of disease that it was causing. You see the bar, the last blue bar, is actually fairly high. And Dothrella was intermediate between the controls and Umbellopsis, but still causes significant disease. And one of the things that is missed here, we actually gained, if you look on the left-hand side, you can see all these different arrows, they're all statistically significant. And on the right-hand side, you actually have disease caused by the same fungus if you have water stress. As you can see that in Dothrella viticola, actually, disease is higher when you have stress. So we were able to show the disease really, that water stress made a difference. And then also for Diaporthy funiculina, the last one, you can see that in the presence of water stress on the right-hand side, disease was higher. So there is intersection here of a pathogen being present and the activity of the pathogen being hastened by the presence of stress. Interestingly enough, Umbellopsis doesn't respond that way. So Umbellopsis is here in the middle, and it actually does well in the absence of stress. And that's probably because it's a zygomyces, so it actually likes water. So in the presence of water, it does well. So this is a little bit concerning, and more research is needed. But again, that was found in a single site, so it wasn't really responsible for overall mortality. In the case of Eucalyptus, I already, here's more symptoms of what we were finding. Again, these were really normally associated with leaf blight moving into the twigs, into the branches. And I already told you that these come from Australia. These are endophytic, and they probably have a positive role. And then when plants become stressed, they become aggressive pathogens like Diaporthy and Dothrella. So the same ecology and biology, but a kind of a different type of disease. And in this case, it was mostly twig dieback and leaf blight. And we had a series of fungi that were isolated in Eucalyptus in some sites, but not all sites. It was the first, all three of these are very interesting because we know about these. So Cytospora, where there's actually, I show you a picture where there was a bleeding lesion. That was Cytospora, and the bleeding is actually the spores of Cytospora. Then there is a bot fungus, Botrosphaeria fungus, Neophysicoccus eucalyptorum. Again, this is unreported in California, and it was causing branch dieback. And then we have these Visconioxia graphostroma that causes charcoal disease. The relatives of the ones that cause charcoal disease in oaks that are stressed by drought. So we also found them in Eucalyptus. These are really, these were more spotty. We only found them in a few locations, and they all are known to have an endophytic stage again. So these are endophytes, and we know they're activated by physiological stress. So what did we, if we have to summarize, what were the potential outcomes? We could have had a primary pathogen like Phytophthora ramorum. We didn't really find any primary pathogen. We could have had only secondary pathogens, opportunistic pathogens. This would have meant that the climate was really driving mortality. Or we could have this third category of latent pathogens. It's kind of in between the first and the second. And so these latent pathogens have an endophytic life stage, and they're triggered by climate. But once they're triggered, you never go back to, you don't go back easily to the endophytic stage. Most likely you remain in this aggressive stage. Not always though. There are some cases where it's possible to go back. But obviously these are not opportunistic pathogens per se. It's a much more complex relationship. And because they already are inside the plant, they have this so-called pole position, and they can cause these very, very rapid outbreaks. So the issue with, I already mentioned it, this is just a summary of what I just said. So acacias don't really belong to California. They were planted assuming that they're really drought tolerant, and speaking to botanists, they tell us they're not drought adapted. They actually come from mesic environments in blackwood acacia in particular from Australia. These latent pathogens, there's been a big body of literature showing that high rainfall facilitates infection of the endophytic stage. So this should light up light bulb, in our heads that what happened in 2017 was likely a major infection of Bay Area trees by these latent pathogens because of the huge amount of rainfall. So that infection probably has been occurring for decades, but it was at low levels. And then because of the huge amount of rainfall, we had a very, very high infection that basically almost every individual, especially with acacias that are densely packed, would have been infected. And then because of the drought, these endophytes become pathogens. Once they're pathogens, they can be very fast, and then secondary fungi jump on these trees and hasten the tree mortality. So bottom line, the weather itself does not explain the mortality. Infectious fungi are actually killing trees. These are not opportunistic fungi, but they're latent pathogens that have a symbiotic relationship with their host for a long period of time. In the case of eucalyptus, again, the same, they're not native, so they're probably off-site. But none of the fungi we isolated are primary aggressive pathogens. We did isolate some latent pathogens again, but these cause a canopy dieback rather than a mortality of the entire tree. And again, the climate drives these outbreaks, but these are of less concern because the fungi that are involved are specific to eucalyptus. And so usually I call these pathogens Dr. Jekyll and Mr. Hyde pathogens. And again, I already mentioned that there is a physiological trigger that makes them become aggressive. And I go back to the last point here in this slide, that originally pathologists have not paid a huge amount of attention to these pathogens because they were thought to be important in marginal conditions. So where you were planting trees in marginal sites that were really not adequate, near roads, for instance, then you would have a lot of these outbreaks by these latent pathogens. What's happening now is that the entire range of these trees is becoming marginal because of climate change. So this is huge. And because of this, because climate change is making the current range of entire tree populations unfavorable to the tree species, I believe the latent pathogens will be major drivers of plant mortality, and they will be reshaping the composition, the structure of our terrestrial ecosystems more than invasive pathogens, more than exotic pathogens. Because these pathogens, well, some of these latent pathogens are exotic, but overall, as a category, they have the better half. They have the upper they're in a better position to cause mortality because they already are inside the tree. So what to expect? So there's going to be high mortality of these trees, and this will cause a large amount of dead wood and debris. And this is unfortunate because these latent pathogens are even worse than we thought. There is a big body of literature showing that these latent pathogens that were symbiotic in living plants without causing symptoms, then they become pathogens, and then they become saprobes, which means they do very well on dead plant matter. So they do very well on the dead wood from the trees that they killed themselves. And what happens on this dead matter? They actually sporulate. So they produce the spores, and then the spores will start a new cycle of infection. So the amount of dead woody debris and also of green debris is particularly problematic because it increases the inoculum potential, the amount of spores that are present in the air. And if the pathogens involved are generalists, like in the case of the acacias, then we put at risk other tree species that are nearby. Okay, so climate change is causing stress. I talked about this. Where are we seeing these large-scale mortality? Well, okay, so we're seeing them on plants that are off-site because they're not native. We're seeing them on manzanitas. Especially because manzanitas are really adapted to particularly dry sites. They do really well in dry sites in general, most species do. These dry sites have become even drier, and they probably have gone below a certain threshold where the manzanitas entered a physiological state of incredible stress, and this activates these latent pathogens. And then blue oaks and gray pine in the Sierra Nevada foothills, blue oaks and gray pine, they grow in the very low elevation where it's incredibly hot. Now it's even hotter. It may simply be too hot for these species. So they were already adapted to very hot climates, and right now it's probably above the threshold that they can bear. The same thing that's happening to manzanita. And we observed the same thing in California bay laurel, because California bay laurel should not be where it is. California bay laurel should be a riparian species, and where it's away from riparian environments, it's now dying because of the drought. But the truth is that even if it's native, that tree does not belong. So am I right when I see these things? Yes. So we did a study on manzanitas in Mount Diablo. These were the symptoms, very large scale symptoms as well. You can see these nice lesions on the manzanitas. These were basically ubiquitous everywhere we worked. Look how beautiful these lesions are. I think they're beautiful. And so this is what's killing the manzanitas. These were the sites where we were working. And out of these sites, in 4 of them, in every single one of them, we identified fungi that were associated with climate change. But in particular, these endophytes, we found them in 4 sites. So these were very prevalent, this Neophysicoccum. And so this actually matches supports our hypothesis. So the fungus we isolated is Neophysicoccum australe. It could infect at equal frequencies 2 species of manzanita. It's already been reported in Central and Southern California, causing large-scale mortality. Then we found other fungi like Diaporthe australafricana and Cytospora austramontana that are also known to be triggered by unfavorable climate. So what's happening here is on the first square, either we're planting trees off-site, or we are, or trees that are native are off-site, sorry, because of climate change. Every time that it rains, the spores of this latent pathogen infect the trees. The trees remain healthy, but then there is drought. Symptoms start to show up. The symptoms cause, they start to grow. They cause dieback. You see, you have the dieback. The plants become weaker. Then you have secondary fungi that jump on. So you have mortality is actually caused by primary pathogens, which we already identified, and secondary fungi. Together, they cause large-scale mortality. On these dead trees, when it rains, you have production of spores, and the cycle goes on. The most important thing is this first box here. This first box could be planted trees, or it could be native trees that have grown out of their natural habitat, like in the case of bay laurels, or trees that are off-site because of climate change, like manzanitas, gray pines, and blue oaks. So what's happening? Why are they being activated? And so I wanted to show you 3 things, and I won't talk about them because the first one is increasing temperature. There is no doubt that there is climate change going on. On the left-hand side, you see the temperature is increasing. On the right-hand side, there's a decrease in fog. So for coastal environments, this is very significant. And then in the middle, what it's happening, you see these curves are all going up. It's the occurrence of extreme events. So basically we have very wet and very dry years next to each other. So this is happening more often. So we have stronger fluctuations, which are not easily dealt with by plants. So these are the three factors that are happening. Now the question is, we know that there is mortality going on in many species of plants, both exotic and native. Are we in case number one, where mortality is going to go up, go up, go up, go up, and reach very high levels, basically decimating these plant species? Are we in a situation like two, where we're looking just at one cycle and then we're going to go back to normal? So we're right on the peak here of the blue dotted line or dashed line. So we see a lot of mortality and the trees will come back. Or are we in a situation like three? And I think three is where I bet we're in a kind of three situation where we are now on the peak of the second curve in the yellow line, and then trees will come back, but then we're going to have another outbreak. And then, as you can see, mortality levels go up, which means that those populations may be decimated, but it's going to take a longer time. The end effect may be the same. Unfortunately, we don't really know if we are in scenario one, scenario two, or scenario three. So the recommendations are a little bit difficult to make. So to conclude, BMPs, the first thing to keep in mind, so the best management practices, is what I'm saying here is based mostly on the literature, because we have very little experience with these fungi in these ecosystems. And these are not all or nothing recommendations. So all I'm saying is doing a little bit of what we're suggesting may be good no matter what. You don't have to go all the way. It may be not cost effective. It may not be possible. But the number one, and the number one thing to keep in mind is these trees are unhappy. Anything that makes them happier is bound to reduce disease levels. And then sanitation becomes important, especially when we have these generalist pathogens or when we want to preserve the local population. So, in the case of eucalyptus, maybe it doesn't matter if there's eucalyptus fungi that keep killing them, unless you are in a place like Stern Grove or a place where you really want to maintain eucalyptus. With acacias, it may be a problem because those fungi can jump on other hosts. With manzanitas, it may be a problem because we want to preserve our native Manzanita populations. And so that debris that's caused by these bot fungi may actually will support more sporulation and causing new waves of disease. So maybe we need to start thinking for the exotic species that they need to be removed. Maybe it was a mistake. In fact, I can say it was a mistake to plant them throughout the Bay Area. So if we have to choose, I would look, based on the knowledge of my properties, I would start eliminating those eucalyptus and those acacias from the sites that are drier, because that's where the effects are bound to be stronger. And that's really where these trees probably don't belong now with the knowledge that we have. If for some reason we want to maintain these tree species, or if I'm working with native species, then removing the weakest trees is actually beneficial for two reasons. One is because these latent pathogens are infectious. And so social distancing among trees, which I've been teaching for 30 years, now people understand what it is. So social distancing among trees is very important in actually reducing contagion. So immediately by reducing the density of a population, you reduce cross-infection by these latent pathogens. And then also you have more water and more resources available for the surviving trees. So that's definitely beneficial if you have to keep these trees. Again, if you care about the species that's growing, or if you have a generalist pathogen, like with the acacias, remove the debris. You can burn it, you can compost it, you can mix it with the soil. So this is something interesting that I know it's something that we don't necessarily want to do. But in some areas, maybe we need to do a little bit of everything here to actually reach enough removal of debris. If the trees are particularly valuable, let's say if you've ever been to Berkeley, we have one of the tallest eucalyptus in the world, right at the entrance of campus. So that tree we actually want to preserve. So that's the kind of tree that we want to water. But again, we have to be careful, because if the water touches the canopy, then that Pseudocidovia will actually be will actually grow more rapidly. And so the watering has to be exclusively in the soil, not in the canopy. And again, if we really want to preserve some of these trees, especially with eucalyptus, one option may be to prune these trees, and also with acacias. So if you have a canker that's on the branch and not on the stem, or if you have a eucalyptus, you can prune the trees severely. And the idea is that you're going to remove the debris, the green waste. You're going to remove the inoculum, and a tree with a smaller canopy will have less of the pathogen, and it will also require less water, and so potentially it may be happier. And so again, I already talked about the individuals. And so again, there's a little bit of there's a difference between acacia and the eucalyptus. And I guess with the acacia, I would also put the native plants, especially the ones that are still in their native habitats, like the blue oaks and the great pines. In the case of acacia, blue oaks, great pines, we probably want to eliminate some of the debris because it could start a new cycle. In the case of eucalyptus, again, they're very host specific, so unless you want to preserve the tree, I'm not particularly concerned. And this is it. This was a lot of information done by very intensive work that happened in about 6 months. And I will take any questions that the group may have.
Okay, there's some questions on the chat, Matteo, so I'll just read them
Sure.
for you. So Nikki Hansen writes, so for the most part, the fungi are beneficial unless the tree hosts are stressed. Is that because the fungi start trying to be more aggressive and taking resources from the tree?
Well, it's actually, it's more like it's not really kind of the way we understand it. It's not a gradual process. It's a trigger, it's an on-off switch. So you go from being beneficial because by protecting the plant, basically this is the evolutionary probably process. The endophytes, by protecting the plant, they also protect themselves, right? Because the plant is their home and they want to be there for decades. So there are some things that these endophytes have been selected to be allowed to coexist in the plant because they provide some benefits to the plant. Okay, and we have done one study in French Polynesia where we have proven that these endophytes protect from aggressive pathogens that come from the outside. So we know that's a huge role, right? So the endophyte is producing something because its machinery is still active, right? The genes are still expressed, and so these fungi are not like dead inside the plant, they're alive, they're expressing genes. What happens is if the environment is not favorable to this symbiosis, then the fungus goes to being a pathogen. So there's no in between. So the moment that the plant doesn't provide that nurturing, favorable environment, the pathogen the fungus turns into a pathogen. So when we look at these latent pathogens, there may be a difference because some of these endophytes may be very aggressive pathogens and some may be not so aggressive pathogens. So there may be some difference there depending on the species, but I don't see this as a gradual process. It's just, okay, now the plant is not supporting me anymore, so I'm going to kill the plant because by killing the plant, I can get the food that I need to survive.
Okay, great. Len Erickson has a question. He's on the coast, and his question is about Quarry Park, Matteo. And Quarry Park is exclusively eucalyptus. It's probably the largest, it's hundreds of acres of eucalyptus if you have ever seen it on a map. So he wanted to know if there's been any studies in an area like Quarry Park where it's just really one variety of plant.
I don't know. We didn't study it. We had to get the results fast, and so we selected the studies based also on the funding agencies that they own them because they really wanted to have answers. So I don't know. I mean, if we follow the hypothesis that we laid out here, is the more coastal these populations are, probably the less amount of dieback there's going to be. because the less, even if we do have less fog days, I would say that the amount of stress on truly coastal areas will be less marked. And so, for instance, in Stern Grove, I haven't heard of a very heavy decline of trees there, or in Golden Gate Park, but in the East Bay, yes. So, there you go. There's a substantial difference. But I think eventually, it's the point will be, I think, to give you a term of comparison, that eucalyptus in Southern California will do better because, first of all, it's red gum and other species that are more drought adapted already. Instead, blue gum is not particularly drought adapted. And so now that we're entering this new phase, which probably will last forever, I mean, thousands and thousands of years, really these trees don't belong anymore. Potentially at some point, it may also be that the truly coastal eucalyptus will suffer. And so, but I predict that it's going to happen a little bit later.
Okay, Dan Dempsey from Woodside Fire has a question. He's seen a dieback in some of the fir trees. Do you think that is just drought related, or do you think this has some correlation?
Well, Doug fir, I'm assuming.
Yes, I think so. Yeah, yes. Okay.
So, there's a lot of stuff that's being reported, for instance, from redwoods. So we know that the redwood situation is very similar. We do have, for Douglas fir, there's a whole series of native diseases that already affect Douglas fir, but I haven't heard of any particular one being dominant. But I can also see how the stress, the drought stress on these trees would trigger the activity. So we have a fairly rich range of native pathogens affecting Douglas fir. So I wouldn't be surprised if that's were the case. It certainly is the case for redwoods, and especially when you have redwoods that are planted, off-site, like most redwoods in the Bay, with the exception of the truly coastal counties. All the redwoods that are planted are obviously off-site, and those are experiencing a lot of these latent pathogens, cankers, so these diebacks. And it's also interesting, there is a series of pathogens that unfortunately have emerged that do well in where you have a lot of relative humidity. So it's like these trees are getting clobbered both by the drought and by the present relative humidity. So it seems, and that's why when I was saying when you water the tree, make sure that it doesn't reach the canopy, because a tree that's stressed because of the lack of water, if you have high relative humidity in the air, you will get some of these foliar pathogens that will really make the tree unsightly, even if it's not necessarily going to kill it. It's going to cause extensive needle defoliation in these trees. So it's unfortunate, because that's an interesting combination. And those are, even if some of those are new, those are, we suspect they're native pathogens, but because these trees are stressed, they take advantage of these trees, and when it's very humid, they flourish. And so, it seems counterintuitive, but that's how the mechanism, is functioning.
Okay, I have a question. So it sounds like you've correlated unhealthy forest in terms of unhealthy tree density as part of maybe the potential stress. So
yeah. So that's true. Okay, that's true for acacias for sure. So the acacia was very convincing, because we know that these pathogens will, those pathogens are aggressive if they really, move from one tree to the next if the trees are growing next to each other. With eucalyptus, it's a little, we know that that's probably happening too, because the major player, but I should clarify that because the major player was Pseudocidovia eucalypti, like the foliar blight pathogen. And because we think that almost every eucalypt already has it inside itself, it's a catch-22. So we can't really, I'm not sure, I think the high density will create a worse situation, because most of these diseases are density dependent. But it can be bad already, even if you have isolated eucalyptus, because the disease will express itself because it's already present. So this is obviously happening. In native trees, certainly with manzanitas that grow fairly dense, that is favoring the spread of these latent pathogens, potentially in bay laurels as well, because they're growing very dense thickets. So I think density is being a big player for these ecosystems. When I talk about blue oaks, it's a little bit more complicated because blue oaks don't grow. You're normally growing in a more kind of savanna type of habitat. So I think we need to understand more what's happening there. That may be more just driven by physiology than by proximity. But I would say that for most plants, density is a factor. And so by reducing density, you should make things better. And also from fire perspective, right? Some level.
So, okay, that's a good segue into many of us do fuel reduction projects. And so we're going to run into these diseased trees, maybe unknowingly. So do you have any specific precautions that we should maybe consider when removing the trees, what we should do with the chips? Is it the same as SOD?
Well, so that's why I was trying to emphasize the difference between the eucalyptus and the acacias. So I feel that, let's start from eucalyptus, right? So I think that in eucalyptus, your prescription should be driven by fire, in my opinion, mostly by fire issues, more than plant disease issues, unless preservation of eucalyptus is key to you, right? So again, if you are in an area where preservation is key, then you're gonna go into the next box. But if you're not in that situation, and honestly, I would, I think that these outbreaks would support removal of eucalyptus, but we have to be careful because they do provide some wildlife. There's some, you can't just, there's some people that like eucalyptus, and there is some beneficial, purpose eucalyptus serve. So again, we want to be a little bit careful, but I would say that debris removal is less of a pressing prescription for disease, more rather I would think about fuel, distribution. For acacias and maybe other native plants, I think the issue is a little bit more complex, because these particular pathogens are not just associated with acacias, but they're very broad. They're very infectious once they're in their Mr. Jekyll, in a Mr. Hyde phase. They're very infectious. And so I think that we need to be a little bit careful with them. And that's why, so they could put, I think the big issue here would be doing nothing. And when you have these outbreaks, could cause a high buildup of debris, which also it's obviously will impact fire behavior, but it will also cause a high buildup of fungal spores. And then these fungal spores are likely to infect the trees that are next to them, even if they're not acacias anymore. So let's assume that acacia are all gone. You have coast live oak. We may see outbreaks of these fungi on coast live oak next to them. So I would say that I encourage, for multiple reasons, I encourage removal. And I think we need to treat the debris as infectious. It doesn't have the same, legally speaking, because the fungi are both Dothrella viticola and Porthifuniculina have already been reported in California. They're considered native or naturalized. We don't have the same regulations that, that are extremely strict, but we should be as careful as possible, with limit and definitely getting rid of this. And again, it's not all or nothing. So if you can only get rid of 20% of the debris, that's still good. I would still do it, because you're still taking out 20% of the potential number of spores. And so in your fuel management plans, if you have acacias or if you have native plants that are showing these symptoms, I would assume that you have the same kind of fungi, unless proven, and so I would say I would work towards a reduction of debris. And we had a meeting where we talked about what you can do. And the classical things would be any composting would help. Of course, removal and putting in a landfill would work. So these are not soil fungi. So they don't actually they will not contaminate the soil. And then I went a little maybe overboard, but in agriculture, they actually bury the debris, below the top layer of the soil. And when they do that, disease in these agricultural crops goes down dramatically. And so as maybe a less desirable option, but if I still would keep it in some areas, it may be a good idea to just mix the debris with the soil a little bit if it's possible. But I don't know, sometimes it's not because the soils are so dry, it's just not really a reality. But so these are the three things. Of course, burning the debris if it's possible. I mean, anyway, so I would say reducing the debris and carefully because of the fact that it's infectious. But again, with that, we don't have the huge, compounding issue that the pathogen is exotic. So these pathogens are probably present throughout California. They're just not present at very high levels. So that's the thing. We don't want to move, a huge pile of debris from site A to site B and leave it there, because moving a huge pile of debris means moving a huge pile, a huge amount of spores. So we want to be careful. So maybe bag it or tarp it, or make sure that it gets buried, whenever it arrives in the disposal site.
Okay, great. Yeah, I think most of our projects would include just chipping in place. Some of it would include pile burning or taking it to like a landfill. So I don't think dumping it from one site to the other is really any prescription that's part of any of our projects. So that sounds like that's a good thing. Yeah,
and the other thing is, remember that the disease in eucalyptus and acacias is happening in different parts of the tree. So really with the eucalyptus, most of the big deal is the foliage and the twigs. So that's really what, the big wood, you don't really have to worry too much about it. And so that already decreases the amount of biomass you have to worry about. With the acacias, it's a little different. It's maybe the other way around. So with the acacias, you could actually leave the foliage and it would be better to, if I had to choose, if you delimb them, take out the limbs, I mean, the foliage could stay, the bigger limbs should be taken out, and potentially the base of the stems as well, or the stems, which may be a bigger job, I guess.
Right.
But so I mean, it's not all the same, not all of the green waste and woody debris is the same, because the different parts of the tree that these fungi, infect.
Yeah. Well, your point is well taken about, is it a wildfire resiliency project, or is it a diseased tree pathogen? So I think that your point there is very well taken. Okay, we have to move on to our next presentation. Thank you so much, Matteo. This was really, I don't think anybody here has heard any of this. This is right off the presses, it sounds like. And
yeah, we just finished like 2 weeks ago. So.
Oh, okay. Okay, great. And if you could keep us updated, I'll try to stay in contact with you. It might be good to present this to a larger group, some of the conservations and some of the arborists in the county. It might
I'll be happy to do that.
Helpful to present. But thank you so much for all your research and continuing to do such really great work.
Okay, well, thank you. Thank you for your support. Appreciate it.
Take care of yourself.
Okay. Bye-bye.
Okay, right. All right, well, we heard it right from the horse's mouth there. It looks like we're in for more dead fuel on the ground. But Matteo will keep us updated. Okay, we're going to go right into our next presentation. And Jennifer, I think you're there, and I have you set up as a co-host. So if you want to share your screen, if you have a presentation that you'd like to show. So I wanted to introduce Jennifer. She's a resident of Portola Valley. She's on the Wildfire Safety Committee of the town of Portola Valley. And Jennifer and various residents and an array of technical experts, they kind of wanted to tackle what they should be doing about some of their southern exposure, their canyons in Portola Valley. And so they did this this year. The report was completed, and it's very informative. And this is not town sponsored by any way. The Woodside Fire District did participate, but this is a neighborhood group that did this, that sponsored it. And so it's very well done. And so Jennifer is going to share the report with us.
Great, thanks Denise, and thanks for having me. As Denise said, this was a neighborhood effort to study canyons and their effect on fire. There are a number of us around the canyon that you see right here, that is bordered by Alpine, Westridge, Cervantes, Minoka, and Golden Oak. A number of us are involved with WPV Ready. I'm also on the fire committee, but as Denise said, really what this was 3 neighbors who were very concerned about this particular canyon and the canyons throughout Portola Valley, and wanted to know how could we increase our fire safety if we're living next to these canyons. A lot of these canyons are very densely forested, and if you look at this property map, you can see that the canyons run on the back of many private properties. So that presents a unique challenge. Just want to do a shout out to my fellow authors, Robert Seidel, who lives on Pine Ridge, which is this little street up here off of Minoka, and Bob Turcott also participated in this study and helped write and take a video that is included in the report. At the end of my presentation, I'll give you a link to the full report. It actually has a number of different sections, and this is just meant to be a higher level presentation. We started with, actually, another neighbor had done a study of the Shady Trail Canyon, which is over up here off the map. Her name was Bev Lipman, and she worked with private property owners to actually clean up the area around the Shady Trail to make it more fire safe. And so her initial work provided a model that we wanted to follow. We hope that this is also a model for other people who live on canyons, and as such have made it an open report that you can add to. And we consulted many experts, and I'll talk about that and get into that in this brief presentation. The common concern was about fire and canyons. There were other concerns that came up on the way that we needed to address, but the primary focus was, how do we make sure that we are fire safe if we are one of these properties, and frankly, well beyond one of these properties, because what we found out. Portola Valley has a safety element that identifies the canyons within town among the highest risk fire-prone areas within the town. Part of the reason of that we found in our study was there are 3 components that are really contributing to these hurricane-like fires that we've seen throughout the state. Wind, of course, is one of them. The embers caused by any type of fire spark is another. And then canyon seems to be playing a role again and again in the bigger fires that we've seen both in Southern California and Northern California. And the reason for that is basically the topography doesn't allow air to come in on the uphill side. So you have, if a slope is steep, you have the air coming from the downhill side, flaming the fire exclusively from that side and pushing the fire into the slope, which heats up any vegetation there and makes it prime and ready for fire. We saw statistics coming out of Australia and other places that had similar slopes about the how the fire spread increases very rapidly when you have a slope because of this preheating of the trees and the brush and the drying out before the fire actually hits them. So you'll see fires that might go, X miles an hour, go 6 times that if given a sufficient change in slope. So the challenges that you run into, I mentioned private property. Since this is private property, it makes any kind of outside funding more difficult. This is something that is the domain of the property owner and the responsibility of the property owner. And the challenge is this is really a new risk to a lot of these property owners. Also, they have different points of view about what's important to them and why they moved to town. We've had decades of fuel buildup. So actually what we're seeing in the canyon now does not represent what we had seen, early a century ago, or when we started moving in the 60s and 70s. That there's much more vegetation. There's not any type of livestock or anything that kind of gets rid of vegetations, and you don't have the same islands of vegetation that you used to have. There's also a big desire in Portola Valley, and I'm sure in other neighboring districts, to preserve habitat and native vegetation. And so we took that into account when we did this study, and also took into account that we're talking about heavy slopes and geologically sensitive soil. So we don't want to do anything that's going to lead to mudslides, which have also happened around this particular canyon. The experts we consulted were Don Bullard, who's obviously the fire marshal. We had a vegetation management contractor, Solomon Pineda from Pineda Contracting, Alex Von Felt, who used to serve on our conservation committee in Portola Valley, but now is and has been executive director of grassroots ecology, helped us with some of the habitat and native vegetation questions. And then we had 2 engineers from Romig Engineering. One was a geologist and one was a soils engineer, to basically do a walk with us through the canyon and talk about fire and fire risk and how to mitigate fire risk while paying attention to habitat, vegetation, slope, and the actual logistics of getting rid of some of the fuel if that's the choice that people are going to make. We wanted to get some common themes about the canyon together so that we can share them with other people on canyons, because we think that some of these themes will show up throughout the canyons in Portola Valley. So our process was to do a canyon tour. We took all of our experts and we viewed the canyon from the bottom end of an overgrown trail from Alpine up Westridge. We couldn't go through the whole trail because it was blocked by poison oak, but Stanford allowed us to come from their property up that trail to take a look at the different vegetation themes that we saw. We also viewed the canyon from above at Pine Ridge, and then from the top end at Cervantes. We put together a video of the canyon tour with the experts. So that's something that's in the report you can click on, and it's a YouTube video that's under a half hour long of us walking through all of the vegetation and talking about the themes that came up, and just basically asking questions of the experts and listening to them answer those questions. So that's available in this report for you to view. We gave each expert after that question and answer form that we filled out with what we felt we learned in our tour, and they edited it pretty heavily in some cases to make sure that we were getting the right messages out. And so those question and answers are also part of the report. So those are verbatim transcripts from our experts on various questions that came up regarding habitat, vegetation, regarding fire risk, and you can get that in the report as well. I'm just going to go through our key findings right now. I think you should think of this report as an open-ended report and something that we're going to currently add to, or maybe before the next fire season, we'll probably have a re-release of lessons learned, because since we released it in June, we've had some questions and are compiling those and plan to do, a refresh of this to keep it current. So the basic findings were around structures. The advice is to extend your defensible space to 200 feet on steep slopes, and we're defining steep slopes as greater than 30 degrees. When Don walked through the trails, he talked about being able to see through all of the vegetation to the other side of the canyon, or to a wall of the canyon. So that involves clearing ladder fuels and raising the lower reaches of the canopy so that there's a vegetation-free gap that exists to 8 feet above ground. When we did our tour, we also had the native vegetation and expert there, and Alex von Felt talked about the first 1 to 2 feet above ground is the place where a lot of that native vegetation lives. And when you can encourage that, you can crowd out more fire-prone and non-native species. When we talk about, fire-prone species in the canyon, we're really talking about, by and large, poison oak. We saw more poison oak than probably anything in our canyon. There are some other themes that I'll get to, but there are also non-natives there, and other plants that come in that are low quality vegetation that can be removed. Islands of vegetation are permissible in terms of fire if they don't provide a ladder to the canopy above. So what we want to think about here right now, if you do an aerial view of the canyon, you'll just see one whole large blanket of what looks like oaks, but there's a lot more there than just oaks. And the oaks alone aren't necessarily the problem. What would be nice is to have, groups of oaks in that canyon more than just a huge blanket. We also learned to stay 25 feet back from creek beds and riparian corridors. We had some question when this report came out of, what is a true riparian corridor and what is not a true riparian corridor, and that's something that we'll address in a future revision. But what, just as a summary, wildfire risk mitigation in these canyons and habitat preservation are not mutually exclusive, and there are ways that we can actually preserve the habitat in a way that will help the native species and help the canyon not burn down, which is not good for any of us. So I talked about the ladder fuels. 80%, we kind of estimated by, after our walk, about 80% of the ladder fuels are made up of dead wood and woody branches, poison oak, Scotch broom, and chamise. Those, if those were just taken out alone, we would go a long way to getting more fire safe in the canyon. There are also a number of target trees in the canyons that we think should be removed. Pine is one of them. Eucalyptus is another that was identified, and bay trees, with a couple caveats on the bay trees. One of the reasons for removing them away from the riparian corridor is they carry the sudden oak death, the vector for sudden oak death. And when they are near an oak tree and you want to choose, we generally lean toward the oak tree and saving the oak tree. There might be a case of a bay tree that's near a riparian corridor that's a heritage tree and terrific and can be kept, but that's one that oftentimes seemed like were either next to a pine or next to an oak tree, and we're not helping the ecosystem there. The two of these trees we've identified in town as the flammable 5, which is pine and eucalyptus, that we're targeting for removal for fire safety reasons. Just a note here about some of the things we found out from our engineers, and you can look at this more in detail in the report, but there is a town ground movement potential map, and you want to check that if you are removing any of these trees from your private property. Some of the instability that we saw in soils was very visual, and we identified it in the video. So you also might want to check that to gather some lessons from that. So these are the report elements that I'll outline. The first part is that canyon tour. We have a video, and then we have the Q&A interviews with each expert. We also included a focus or a perspective piece from Bob Turcott, authored that piece on creating sustainable habitat sensitive fire solutions. So this is the piece that really goes into the fact that if you pay attention to habitat, you actually can end up not only protecting your property against fire, but coming up with a system that is easier to maintain, less expensive, and solves a number of problems at once. We also put in here a vegetation management contractor list. This is for Portola Valley residents, but also they work outside Portola Valley. So in larger San Mateo County, this would be relevant. And we took contractor instructions, and these we spoke a lot to the San Mateo County Firewise group. So thank you to Sheena and her group on that. And we took them and to see the canyon as well. One of the things that San Mateo County provided us were contractor instructions that they had put out to bid for San Mateo County parks to maintain those parks in a fire safe way. So we looked at those guidelines and translated them based on what we had learned to what would work for our canyons. And then we translated that into Spanish as well. So that's just meant to be provided to give residents something that they can give a contractor to discuss with the contractor about things like expense and the best way to do removal or chipping in place or that type of thing. So our next steps on this is we just want to share this report with others, and I will give the link in the chat after we complete this, but I'm calling it an open source report, just borrowing a term from technology, because this report is currently being updated. This is something we put together after a few months of study, but we have already found out things that we want to look into today. For instance, the whole issue of funguses and disease trees is very interesting and something we would probably look at and identify as something we want to address just in a Q&A format or something like that. Others can use this report kind of as open source material and add to it. I think that would make the collective wisdom of the crowd better and very helpful. I know that our conservation committee is doing something that we're looking at now and how we can add this to the body of knowledge of this report, which is an understory study. So what do we do about the plants that are in the understory? And it very much foots to what we've studied here, but there's just a little bit more visuals and different lessons that they wanted to get across. We've already distributed it in the Westridge WPV Ready block captain group. And we want to extend the lessons to other canyons throughout the WPV network. We've also already presented this to the fire committee in town and are looking to extend that across other parts of the town. So we wanted to come in and speak at this, and thank you, Denise, for allowing us to come and speak, just to get out the message about canyons and to give everyone a little bit more information and benefit of what we studied, and are looking for other ways to get out the message. And we are also starting to work on exploring funding opportunities. And I think I indicated at the beginning, it's a little bit tricky because it is private property. And what we've identified is trying to think about people who have lived here for a long time, who might need some help, who can't do the vegetation removal on their own, or, you know, maybe on fixed income. And so we're looking at some of those funding opportunities and seeing what we could do there. I think that is about it.
Okay, thank you, Jennifer. I mean, the report is very, very thorough and well done.
Thank
I have a
you.
question, if you don't mind me asking, was there a cost for this report?
So we were really, really lucky. We benefited from, you know, those experts provided their time for free, all except for we did hire the soils engineers and the Romic folks. So, you know, that we did pay them for their time, but otherwise it was just a volunteer effort.
Great, great. Well, I mean, I think the fact that you're sharing this with everybody for other neighborhoods or communities to use it as a template, and they can pull in their neighborhood resources, they can pull in their CAL FIRE resources, RCD possibly, if it's, you know, in a riparian area that, you know, they're really great about that. And then you're right, I think the funding, once you develop the project, I think then the funding to how to get the project done, how to do the environmental review becomes kind of step 2 and step 3. And that's where FireSafe is hoping that, you know, we will be able to eventually find some money for homeowners. We get asked every day, yeah, you know, how to do fund projects on private property. So, but I think this is really great that you guys created this. You know, you saw the need and you wanted all the information, and now you have it, and potentially you'll get town support, or, you know, maybe some support from another entity to help you, you know, finalize the implementation.
I think so. I mean, I think we're kind of in an unusual place because we are just a group of neighbors, right? This is not done from any kind of, you know, from a committee position or anything like that. And so we're trying to kind of work through all the organizations that have an interest in this. And so I think probably, you know, the town will be next, and hopefully we can get some support throughout and get that message out through other, you know, agencies.
All right, well, I put the report, I sent out the report via email for the meeting invite. It's in the chat. The YouTube video is in the chat. Sarah from CAL FIRE added some resources in the chat. So hopefully people are reviewing the chat and extracting what they want. So